Okay, well, thank you. Welcome. Hopefully, you're all maybe doing a lunch and learn now, eating your lunch and watching this very interesting talk on macroalgal blooms. So, what this talk is going to represent is just really kind of a synthesis of ideas of trying to make sense of a group of algae. So, algae is not an official taxonomic term, it's kind of a lumping term for different types of photosynthetic organisms, whether they're prokaryotic or eukaryotic. So this talk's going to be focusing on macroalgae, and I'm going to explain what macroalgae are in freshwater ecosystems. So this is an iconic photo from a few years ago that there was a feature in National Geographic magazine. And normally when I show this picture in person, I kind of ask people to tell me where this photo was taken. Sometimes those of us who live in North America think when we see a massive algal bloom like that, we think, oh, it's got to be Lake Taihu in China or somewhere where there's lots of nutrient pollution. But in fact, this is one of the Great Lakes, Lake Erie. And I believe this photo was taken in 2012. But of course, every year over the last almost decade, Lake Erie experiences these phytoplankton blooms. So these would be microalgae. In particular, this is a cyanobacterial species known as microcystis. But what is this? And so when people look at this, and I'm the first to admit that when I first came across this organism in the Rocky Mountains, clear waters of the Rocky Mountains, it was basically had built up along the river's edge, and it looked like used toilet paper. And of course, I wasn't the only one who thought that. We were getting calls of, was there a sewage problem in the Bow River? And sure enough, at that time, when I looked under the microscope, it was a diatom. And this is Didymosphenia geminata. We usually refer to it as Didymo. Some people call it rock snot, but we're trying to move away from that moniker. But it looks like shag carpeting. And so most of what you see here is actually the stock material. And so this photo, in fact, was taken by my friend and colleague, Caroline Gillis, who's in Quebec. And in northern New Brunswick and Quebec, in the Restigouche watershed, this photo was taken. And mostly what you're seeing is stock material. And so it's really just the slight golden hue you see are the cells themselves. And then finally, here's another photo of benthic algae. It's mostly Cladophora. Of course, there's going to be other species in the mix, but it is dominated by the filamentous Macroalga Cladophora that forms these blankets the benthic zone in nearshore areas of the Great Lakes. So this is a photo from Lake Michigan from a few years ago. And once Cladophora kind of reaches its peak growth, typically it'll slough off either through wave action or just natural sloughing events and will build up on the shoreline. So it's very common in Lake Michigan, Lake Erie, and Lake Ontario for this wall of green to form that's mostly Cladophora. So who are the freshwater macroalgae? Well, I've pinpointed kind of the three main contenders for blooming macroalgal species. So Didymosphenia geminata, Cladophora glomerata, and Nyctalopsis obtusa, which I'll actually spend the second half of my talk focusing on because it's a relatively new invasive species in the Great Lakes Basin. But the freshwater macroalgae, just like microalgae, are diverse. They're a polyphyletic group of algae that are visible at their macro scale. And so that's basically how they're differentiated. Can we see them with the naked eye? And another kind of similar feature that characterizes macroalgae is they tend to be colonial and mat forming. So they grow together and form these dense mats or colonies. And so where do these freshwater macroalgae live? Well, they actually live in diverse habitats. There's not one habitat that is ideal. You can find macroalgal blooms like Didymo in very clear, low nutrient waters. This is a photo of Bow River near Banff National Park that I took a few years ago. And here's another picture I took along the shores of Lake Ontario, where wherever there's hard substrate, Cladophora loves and grows really well. And then finally, this is a photo of Lake Scugog that shows macroalgae just below the surface. So charophytes are the main macroalgal species, because I probably should have mentioned this before too, is that macroalgae are not aquatic plants. They are algae in the sense that they aren't vascular plants with a root system and kind of the more complex structure that higher plants have. Okay, so similar to phytoplankton blooms, the incidence of macroalgal blooms has actually increased over the last 10 to 20 years as well. And so that raises the question, are the ecological drivers, so the ones that we know are important for driving phytoplankton blooms or macroalgal blooms, are they the same for macroalgae? So when we look at the evidence, there is limited evidence for climate-driven blooms, although there has been some work. And there's been increasing evidence that nutrients is not the main driver of the blooms. So for example, there's been some work, in fact, in John Small's group, they published a study about 5 or 6 years ago that showed a strong correlation with the increased incidence of didymo blooms with climate, so increasing temperature over a long period of time using paleolimnological techniques. Scott Higgins several years ago did a modeling exercise to look at the role of climate change in Cladophora and found very weak evidence to support a connection. And then finally, there's been some modeling done to try to understand Nyctalopsis obtus's increase in invasion into inland lakes, and there seems to be a niche shift where Nyctalopsis is moving northward. But that's basically it with respect to the role of climate. Now, when it comes to nutrients, this is actually quite interesting because, of course, there needs to be enough nutrients to support biomass. So, it's difficult to have such high levels of biomass that we associate with blooms without there not being enough nutrients to support growth. And so this is very different from phytoplankton blooms because there's lots of work that's being done to show a direct relationship, particularly with phosphorus, in driving bloom level abundance. So the question is then, is this a nutrient biomass paradox? Can we have such high levels of biomass with relatively low nutrients? So I'll just talk about a few examples with Cladophora and with didymo, and the first one being Cladophora, where Cladophora has a long history in the Great Lakes, back in the 1970s and 60s, going back even to the 60s, when sewage discharge into the Great Lakes, particularly the lower Great Lakes, so including Michigan and Erie and Ontario, there was just really high levels of phosphorus from detergents and sewage being discharged into the Great Lakes, and associated with that were microalgal blooms, but also Cladophora blooms along the shoreline. So with the control of nutrients, so in about this period here, sewage treatment plants in the Great Lakes were retrofitted, and secondary treatment was kind of built into the treatment process. And so total phosphorus dropped dramatically. And so by the 1980s, 1990s, you can see, assume here this is Lake Erie, this is Lake Ontario, Lake Huron, and Lake Superior. Lake Erie being, a lot more variable in the 2000s, but overall a dramatic drop in nutrients. And during this time, I'd say in the mid-80s, the literature basically says Cladophora responded to this. Cladophora became less of a nuisance. It was still around, but it wasn't nearly as problematic as it had been. But then by the 90s and 2000s, Cladophora emerged again, and people were scratching their heads, wondering, why is Cladophora becoming a problem again when nutrients aren't nearly as high as they were back in the 60s and 70s. And so one big part of this puzzle was the involvement of a major ecosystem engineer in the Great Lakes, which are the Dreissenid mussels, primarily zebra mussels. Now there seems to be a shift toward quagga mussels, so I'll just kind of lump them all in as the Dreissenids. And famous work was done back in the early 2000s in Bob Heckey's group, where they're able to show that as allochthonous nutrient loadings were coming into the nearshore zone, of the Great Lakes, rather than the nutrients with upwelling events and so forth being kind of dissipated and transported into the pelagic zone, there was this nearshore shunt of nutrients that was being trapped by the filtering activity of the dry-seeded mussels. And so basically what was happening, there was this really tight coupling of nutrients being trapped by zebra mussels, and Cladophora growing on the shells of zebra mussels had a very nice, efficient way of obtaining nutrients in the benthic zone. However, more recent work by Todd Howell and others at Environment Canada, Todd's at Ministry of Environment and Conservation and Parks. I have to always remember their name change. But basically, Todd's been working on Cladophora for decades, and he always scratches his head at, why such high levels, high biomass eruptions in Lake Ontario in particular, when nearshore phosphorus concentrations are very low. So if we're looking at total phosphorus and dissolved phosphorus, so think of dissolved phosphorus as being really the bioavailable form, organic phosphates and inorganic phosphate. And these levels are quite low, so we would even consider below 10 micrograms per liter as oligotrophic. And if you look on the x-axis here, this is just representing, near the shoreline and going down in depth to about 25 meters at sites in Ajax, Oshawa, and Cobourg. And so, yeah, there's something funky going on in Ajax here at depth 8 to 12 meters, but basically there's no significant difference in nutrients by depth in any of these sites, particularly dissolved phosphorus. But there's clearly, Cladophora with lots of variation at each of these sites being highest. This is likely due to light availability driving, being an important driver of growth here. But there's no connection between nutrients in Cladophora. Now, of course, nutrients, it's not to say nutrients aren't important, they are, but it's this common measurement of water column nutrients, which we all do as part of our water quality monitoring, is just really not explaining Cladophora bloom dynamics and growth dynamics. So there has to be something more going on, and perhaps it's the scale that we have to be focusing on. Similar with Didymosphenia geminata, when I did work in my postdoc before coming back to Ontario, I was able to clearly show that blooms were related to low nutrients. So an increase in phosphorus actually resulted in a decrease in Didymo biomass. And this was also supported by Spalding Elwell when they did a systematic review of the literature and found that, whenever blooms were reported, it was typically happening at very low, like ultra oligotrophic conditions. So this is just kind of building up this narrative of, macroalgae are quite different from microalgae in the fact that their growth doesn't necessarily relate or is necessarily associated with nutrients in the water column. So what is it about macroalgae that perhaps could be driving bloom level growth for a particular taxa, such as Didymosphenia and Cladophora and Nyctalopsis obtusa? It makes it very difficult to lump them because they are very diverse in their morphology, their phylogeny, and their habitat. Each one of these species lives in very different habitat conditions. But they do have some key similarities that may provide some insight into why they can, grow to such high levels and basically smother other species of macroalgae. And so one common characteristic is this colony matrix architecture. And so it's just basically a fancy way of saying, they grow really tightly, but, so the cells are microscopic, but their architecture, when they all grow together in this matrix, they form this very large mat. And does this give them an advantage for acquiring nutrients, for example? They also live very close to the benthic zone or the near bed habitat, because it's important to point out that Nyctalopsis obtusa, even though it looks like an aquatic plant, it's a charophyte and it doesn't have roots, but it does have rhizoids that dangle over sediments. And it can position itself right above the sediment, be right in the sediment-water interface. And that's true for Cladophora as well, though Cladophora needs hard substrate to attach to, as well as Didymo. So one way of getting at, what is it that potentially could be driving macroalgal blooms is really kind of zooming in at what's happening at the micro scale. And so this is the role of the epibiome. So this is basically a term meaning on top of, so the microbiota that are growing on top of the macroalgal cells, because they're large. So this is Didymosphenia. This is a really large, robust diatom cell. So I'd say this is about 80 microns in length, 80 to 85. And then you see these little filaments, that's actually cyanobacterial filaments growing off of, being attached to the Didymo cell. And then you can actually see other epiphytes of smaller diatoms with their stalks growing on the large stalks of Didymo. So in fact, it gives you a really good comparison here to show the scales, how different they are. And so when you have microbes, including even metazoans and so bacteria and smaller microscopic algae, they can grow basically, the microbial loop can occur here without any real connection with the outer water column. And this is an area that needs to be explored more. So really the epibiome may have a facilitating role in nutrient acquisition. So if you imagine that the phosphorus is really low out in the outer water column, but here it's tightly coupled and it's just cycling back and forth between organisms, that could be a potential mechanism for supporting growth of benthic mats like Didymo. So here's some evidence to show, at least support, that this could be happening with macroalgae. This is a paper from a few years ago by Vada Bunker in 2006. And this group of authors compared phytoplankton and periphyton. So periphyton growing in the benthic zone in the littoral area of lakes from Quebec, so a group of lakes from Quebec, from the US, and Greenland. And so if you just focus here on the phytoplankton, so these are, this is the phytoplankton microalgae that are living in the water column. And they found in all the lakes, of course, the slopes are different, the different groups of lakes. I suppose the Greenland lakes are have a much shallower slope, but overall, a line was forced here, positive slope, showing a relationship with the amount of phosphorus drives an increase in phytoplankton growth. But when you compare with periphyton, so periphyton growing on hard substrata, so think of epilithic periphyton, versus periphyton growing on sediments, so epipellic periphyton, there's no relationship. So why would there be a difference between periphyton growing on sediments and periphyton growing on hard substrata? And so really the conclusion here is that periphyton growing on hard substrates like rocks depend on water column TP, and you actually see that here. Whereas periphyton growing on sediments are not dependent on water column TP. And so they are likely getting their nutrients directly from the sediments. And so this is very possible what may be happening when we see macroalgal blooms. So as nitrogen and phosphorus loadings decrease with cultural eutrophication, so this is happening. This has happened in Lake Ontario, for example, actually across all the Great Lakes. Although there's been changes in how the Darcinid mussels have shifted nutrient availability, but really, point source nutrient pollution is in decline, and so we're having this decrease in lake productivity. And so is the benthic zone an important place for subsidizing bloom level growth? And we seem to have some evidence of this with Cladophora, for example. So overall, when we're thinking about macroalgal blooms, knowing that taxonomically and phylogenetically and even morphologically, they can be very different. But if we focus on what they do share in common, so that matrix architecture, as well as their proximity to sediments and growing in the benthic zone, we can look at different scales of influence. So looking at, whether it's temporal, so just having pulsed events happening where nutrients become available at the local scale, all the way up to the regional and interannual scales of space and time. But really, it's important to acknowledge that climate change is important in driving nutrient loadings into lakes, for example. Warming temperatures will absolutely influence growth rates of different algae, macroalgae that perhaps benefit from increasing temperatures. Then as you go down to the lake ecosystem scale, the trophic state of the ecosystem is going to be important. So there's going to be oligotrophic lakes versus meso to eutrophic lakes. And then finally, down at the scale of the mat and cell, there's likely going to be a role of the epibiome in facilitating nutrient availability, as well as benthic nutrients being an important source to subsidize growth when water column nutrients and loadings are low. Okay, so now I'm going to zoom right into Nitolopsis obtusa, which is also known as starry stonewort. It's a charophyte that my lab group has been studying now for the last 4 years. Tyler Harold Lyle is my PhD student who started off studying starry stonewort with the Scugog Lake Stewards back in 2016 as an undergrad, and now he has a PhD project that's grown out of the really cool work that he's been able to do and learn. So for those of you who aren't familiar with Nitolopsis, it's a charophyte, but what's really neat about Nitolopsis is that it's dioecious. So it has female and male individuals in its populations. But what's really interesting is as it is a non-native invasive species to North America, only males. So there, if you can see here are the little red antheridia, so the male gametes. So there's only males in North America. Here's an example of Eugonium. We're hunting far and wide to try and find any examples or evidence of females in the populations, but so far we're coming up with just males, which means that only asexual reproduction is happening in North America. So this is what it looks more at the macro scale, living in the benthic area of a lake bottom. And here's a bulbul, hence the name starry stonewort. So it has a star-shaped bulbul that is probably very important in its reproductive capacity. So it is a reproductive organ for asexual reproduction. And here's the fishing line rhizoids that serve to, they're also involved with propagation of starry stonewort, but certainly they're important in, rhizoids in general are important in nutrient uptake in the benthic zone. Okay, so Nitolopsis obtusa is native to Eurasia, so it's native to the British Isles, through Europe, China, and Japan. And in fact, which is not uncommon with many invasive species, it is in decline in many parts of its native range. And this is of concern to those areas, particularly in the British Isles. Nitolopsis obtusa is an important part of food webs. They have native fish that feed on it, live in it as habitat. And so it's actually classified on their red list as being a species of concern, threatened and endangered. But back in the mid-70s is probably the earliest report of it being found in the St. Lawrence River. And then by the 80s, it was found in Lake St. Clair. And so here's Lake St. Clair here, so you can see better. And so not surprisingly, as it's invaded inland lakes, so wherever you see the black shading, these are verified areas. I would say it's probably, there's probably more lakes than are depicted here, but certainly it gives you an impression that where it was found in the Great Lakes certainly is being probably the ground zero for where it was then spread into inland lakes. Okay, so this is what it looks like. It likes clear, clean water. I wouldn't say it likes oligotrophic waters. It probably likes mesotrophic, so medium productivity lakes. But as starry stonewort grows up from the sediments, it can, really virtually reach the surface of a lake. And as you can see, it just completely smothers any other aquatic plants or macroalgae. So in Lake Scugog, for those of you who are at least familiar with the Great Lakes, this is where Lake Scugog is here. It's technically a reservoir, but it's a rather large, shallow reservoir that's been in existence since the early 1800s when it was dammed for a grist mill that was initially built. But it's an important headwater lake system to the Trent Severn Waterway, which connects Lake Ontario to Lake Huron. And here's the Oak Ridges Moraine. So it actually receives very high quality water down here, but largely is an agricultural watershed, as you can see here depicted with the yellow. So Lake Scugog as a shallow lake with high fetch, so it's polymictic, lots of agriculture on the landscape. It's been a eutrophic lake for most of its existence, and in fact, there's been times where it probably was into the hypertrophic range. But definitely a weedy lake. So Lake Scugog wasn't really a destination for swimming, but certainly great fishing. At one point, it was in the top 10 fishing destinations in Ontario. Over the last few years, things have changed a little bit, and it may be due to the invasion of starry stonewort in the lake. So the Lake Scugog stewards first noticed it in 2015, starry stonewort, Anaptusa, if I want to be scientific about it, and noticed that the other invasive species that was always a bit of a nuisance, Myriophyllum spicatum, also known as Eurasian water milfoil, had collapsed and seemed to be replaced by starry stonewort. So really it was the Scugog Lake stewards who were the first to bring this to my attention. And I know around the same time I had contacted Brian Yin at the Lake Simcoe Region Conservation Authority, and he had also found it in Lake Simcoe. So it was kind of fortuitous timing, and so really that was kind of the start of, okay, well let's look at it in Lake Scugog to try and get a handle on what its distribution is. So these are our sites. There's a lot of concern about its impact on fish habitat that was basically being reported, at least anecdotally, in the United States, such as Michigan and New York State. And so this is our opportunity in Canada to find out what impacts it's potentially having. So Tyler conducted, starting in 2016 with his undergrad thesis work, and then now, last year was our last year, so we have 2019 data that are still being processed. But overall, there was clearly a change. So 2016, who knows when starry stonewort first came to the lake. I think, I don't know if Jenny Carosi and Josh Steenfond are on this, are in the Zoom call right now, but we definitely want to figure this out. We want to find out when starry stonewort first came into Lake Scugog. But certainly back in 2016, there was probably, basically equal abundance. So this was a drought year. So there, algal productivity overall was relatively low in 2016, but they're about on par. And then through time, you see a shift where Anaptusa is becoming the dominant invasive species, macrophyte. So I'll use the term macrophyte because it's an umbrella term for macroalgae and aquatic plants. And because they grow in the same habitat, they are competitors with one another, and certainly Anaptusa has become the winner in recent years. So over this time period, we were able to document that Anaptusa was increasing in Lake Scugog significantly. This is a log scale, just to kind of, there's lots of noise and variation, but certainly a positive trend. And interestingly enough, we also documented an increase in dry-seeded mussels. Now, in the past, no one had studied zebra mussels in Lake Scugog, so unfortunately we don't have data to compare it to. But talking with the Scugog Lake stewards, who are kind of very intensely aware of the goings-on of Lake Scugog over the last 20 years or so, they knew that zebra mussels were in the lake, but no one ever really reported them being very problematic. And the same is true for Kawartha Conservation, the lake monitoring agency that monitors the lake. Definitely there, but probably because of the soft substrate, There's not a lot of hard substrate for the zebra mussels to attach to, so they weren't considered to be problematic. But their numbers started to increase over time, and here there's, it's not a straight line, but certainly compared to 2016, zebra mussels, and there are quagga mussels, very few of them. So this is mostly Dreissena polymorpha, which is zebra mussels. And then also, coincidentally, microcystis started to bloom in the lake. So in 2016, it was, biomass and abundance was very variable over this year. But, and it's kind of surprising because this is a dry year too. You don't usually expect to see microcystis blooms in a dry year. And then over time, it also started to increase. And why this is of interest is because even though Lake Scugog is a eutrophic lake, and you think, well, of course you'd see algal blooms when there's lots of nutrients available, but it's not the typical lake that you would see cyanobacterial blooms in. So it's polymictic, it's shallow, it's weedy. And as far as the Scugog lake stewards were concerned, there really hadn't been any reports or problems of particularly microcystis blooms. Like filamentous algae certainly can be periodically problematic, but microcystis was a whole other type of algal bloom that they just weren't familiar with, and it was quite shocking, and it actually made some news headlines. And so because we were able to collect data about the macrophyte community and the macroinvertebrate community, as well as the phytoplankton community and water quality, we were able to really ask the question, is there a role for both the zebra mussels and Nitolopsis obtusa in driving these microcystis blooms? And so I'm not sure, hopefully nothing's being, I have a little bit of the pictures of people on the Zoom call are covering it up, so hopefully it's not covering it up for you guys. But Tyler conducted some structural equation modeling because we thought this would be kind of the best way to tease apart in a very messy system the direct and indirect drivers of microcystis biomass. And so the overall model is statistically significant, but only explains about 27% of the variation. But what is interesting is that the usual suspects come up when it comes to the abiotic drivers. So heavy rain events, there's a positive driver of microcystis abundance, increasing temperatures, which we see in other studies, as well as chloride. And maybe for some of you may not be that familiar, but microcystis is very tolerant of high chloride concentrations. And in fact, in my lab in coastal wetland study, we're finding that in wetlands that are heavily urbanized and have higher chloride concentrations, there tends to be microcystis blooms. So this also made sense. Now, I'll draw your attention to the biotic drivers. And what we're finding is that Nitolopsis obtusa was the only biotic driver of microcystis. And so normally in other studies, especially in Lake Erie, there's typically a tight relationship between microcystis blooms and the abundance of zebra mussels. But in this case, we found that really it was N. obtusa was positively associated with driving zebra mussel abundance as well as microcystis abundance. But there was this kind of very slight negative effect of zebra mussels, but it wasn't statistically significant. So, the statistically significant, sorry, I should have mentioned that, is where you see the asterisk, because really the drivers that came out as statistically significant in the model, so explaining the whole model R squared value. So really the take-home point I wanted to address is that at least we're getting a signal of a potential role for N. obtusa in driving microcystis blooms in this large shallow lake system. And of course, we want to be able to see if this is happening in other lake systems as well. So this has been recently published. I don't think certainly the preprint is available at Canadian Journal of Fisheries and Aquatic Sciences, but if you wait a couple of weeks, you'll get the final proof will be available online. Or if you want, you can email me and I can send it to you. Okay, so just to finish things off here, last summer, we were really lucky that actually 2 graduate students in my lab did a very intensive field survey last summer and now are kind of working up the data. But 60 lakes candidate lakes were selected in this region of Ontario. So for those of you who are maybe not that familiar, so here is Lake Huron, this is Georgian Bay, Lake Simcoe, Lake Ontario, Lake Scugog's right here. But we wanted to know, at this point, all we really know is that Lake Simcoe and Lake Scugog and a few lakes down by Presqu'île Bay have starry stonewort. We know through cottagers' reports that there's all throughout the Trent Severn, there's starry stonewort. But we wanted to get an idea of what the full distribution was based on water chemistry. And so this is an overlay plot of the underlying bedrock. And this zone is known as the land between because it's the land between limestone-dominated bedrock and the Canadian Shield, so granite and other metamorphic rocks. And I'm definitely not going to waste time going into explaining, but this is really neat because places like Bancroft over here, here's Kingston down here, which is on limestone. But this boundary here really allows us to capture broad chemical gradients of lakes. And so they become, we really can create kind of a treatment design along a gradient to test hypotheses. And so the main hypotheses that we wanted to see if there's constraints based on the calcium concentration available to starry stonewort. So Nyctalopsis, just like other charophytes, require calcium and other cations for their crunchy kind of matte architecture that gives their cells strength and body to be able to grow and form this kind of almost like barbed wire architecture. And so with help with, I don't know if Bree Edwards is on the line, but she shared some calcium data that she had for her study lakes from her PhD. We collated data from Kawartha Conservation and Ministry of the Environment, Conservation and Parks. And we were able to capture a really broad range of 60 lakes. So here, based on the literature values, is where the optimal conditions are, so between 20 and 40 milligrams per liter of calcium. But we also wanted to capture lakes that are below that threshold and above that threshold. And that basically being the hypothesis that we'd expect to find starry stonewort in these lakes and not so much in these lakes. Now, I meant to put an overlay of the 60 lakes. I'm sorry about that. But basically, there's lakes here over by Owen Sound, lakes here by Guelph and Kitchener. And the bulk of the lakes was through the land between all the way up through past Huntsville and Parry Sound, just to get those really lakes that we would consider, far enough away from kind of this hotbed here, but with, water chemistry that is, on the acidic side of the pH, on the pH scale. And what I want to draw your attention to, and so this is the density. So I'm not sure if you can see if it's being blocked off here, but this is low densities, color shading purple up to really high density of Nyctalopsis obtusa. And probably the first thing you're going to notice is that, that's the Trent Severn waterway. And of course it is, but we didn't sample in the Trent Severn waterway. All of our candidate lakes were isolated from each other. So they weren't connected to the Trent Severn, they weren't connected to each other. And we can clearly see that potentially the Trent Severn waterway is a major source for, with boats being moved around and, perhaps birds. We're basically finding Nyctalopsis obtusa everywhere, even in places that we wouldn't have expected to. Now, it doesn't seem to be particularly happy in the lower calcium lakes, but it's there. So I just wanted to kind of give that little tidbit. Right now, Tyler's working on doing niche modeling and other types of models to try to understand more what's driving abundance in the different lakes. But I thought that was just a nice kind of visual representation of how prolific and abundant and obtusa is throughout cottage country in Ontario. So without further ado, hopefully I didn't go too long here droning on, but I definitely want to acknowledge Tyler, who has done all of the work on Nyctalopsis obtusa, and our field crew. Not all of them are here, but there's Claire, Eric, and Aaron. These are, Eric just defended his master's, and Aaron's a PhD student. And of course, the Scugog Lake stewards, and I'm noticing that it's probably Barbara, who's on this call, took this picture, that she should be prominently in this photo. And if you have any questions, feel free to contact me through my email or website. And I'll leave it at that and entertain any questions.